Welcome to this NOBIC talk, NOBIC micro talk, and we have been running this for a few months, and over the time, this has become sort of a prestigious event where the best of the best got to speak. So we had Yongri speaking, we had Esther speaking, just for example. And since I am the one who organizes it, then there was nothing to prevent me from joining this illustrious lineup. So that's to explain why I'm here today. And otherwise, why the topic, why I'm going to talk about FCS, which is quite logical because my research was mainly revolved around FCS before I started working in microscopy imaging facilities and got involved in a variety of other microscopy-related stuff. And I call this talk From Noise to Knowledge, which is a paraphrase to the name From Images to Knowledge, which is a image analysis, image processing conference, mostly focusing on open source tools like Fiji, Napari, and so on. And so I use this opportunity to promote this because they have all the talks and workshops available online. So if you are interested in any topics in image analysis, this is a good place to go. And there's also, there was a workshop on FCS, on imaging FCS in May during this year's Images to Knowledge. So you can find it either there, or you can also find it on NOBIC YouTube channel. And let's start now with the topic for today's talk. So we start with some noisy images or noisy data, and we want to derive some useful knowledge from them. So if we have any microscopy data, microscopy image, so the noise will of course include instrument noise, such as camera readout noise, detector noise, and that is not very useful unless you want to characterize the performance of your camera. More interesting source of noise is blinking of fluorophores. Of course, if we're talking about a fluorescence image, so the fluorophores can switch between a bright state and a dark state, and that will add to the noise because the effective number of fluorophores which are contributed to the value in each pixel is changing if they are switching between bright and dark state. So this is interesting if you want to characterize properties of your fluorophores, but even more interesting are fluctuations in local concentration caused by the movement of the molecule. So if the fluorophores are not immobile, if they are attached to molecules which can move around, then there will be also fluctuations in the number of fluorophores locally caused by their diffusive movement. And this is quite interesting because if we study this, we can learn quite a lot about our sample. We can learn about the mobility of molecules, or I should say fluorescent particles, because they don't have to be just molecules. Those can be also some complexes of molecules or even larger particles, maybe virus particles or liposomes. We can also learn about their concentration. And if we know their concentration, we know the overall intensity of fluorescence, then by dividing the intensity by the number of the molecules, we can arrive at their brightness. So this is, again, could be interesting because if you have molecules labeled by a fluorophore. So if the molecules, for example, form aggregates, then those aggregates will have higher brightness than the individual molecules. So that can tell you about processes like oligomerization, and of course, and all this can be used to study in general all sorts of molecular interactions. So I use a very vague term here because the range of the particular interaction that can be studied is very broad. And now we have a fluorescent particle which is diffusing, and we are looking at some small observation volume. And if the particle moves through that volume, we will register a burst in fluorescence intensity. And for successful application of the study of the noise and translating it to useful information, we require following. It's good to have a very small observation volume from which we detect fluorescent signal, and or a very low concentration of the fluorescent particles. Why is it important? Well, because we are interested in the fluctuations. So imagine you have a hall full of people. There are maybe hundreds of people there. So if one or two people go away or come in, it doesn't make a big difference. Well, on the other hand, if you look at a room where there's just one person, so if the person goes out or another person comes in, that makes a huge difference in the number of people. So that if there's a one person, the one person goes out, that's 100% difference in the number of people. So similarly, in this case, if there's a low, if the volume is small and the concentration low, so we have very low number of molecules inside of the observation volume. So we can see very clearly the changes caused by the diffusion of individual particles. Standard realization of that is in confocal microscopy, where we have our confocal detection volume, which could be about 1 femtoliter or less. So that's very small. It's a diffraction limited volume. We can make it even smaller, but that requires quite fancy setups, like using STAD or using some near field optical effects. Then we require single molecule sensitivity, because the fluctuations come from the individual fluorescent particles. Of course, we can, if the particle is labeled by many fluorophores, that might relax a bit the requirements on the sensitivity. But we have to be able to register the fluctuations caused by each individual particle. So again, in a confocal microscope, this is usually satisfied. We have the sensitive detectors, photomultipliers, or even better, avalanche photodiodes and similar detectors. And we require sufficient temporal resolution, because we need to capture the whole the burst, and we need to measure the duration of the burst. So we need to sample the burst at several points to be able to know whether this is a single burst or whether there are multiple bursts. So typically we require something like at least the resolution should be at least 10% of the typical diffusion time, of the residence time of the particle in the observation volume. Again, this is satisfied in a confocal microscope because the time resolution is very fast. So in a way, a confocal microscope, that is a standard FCS instrument and has been used for FCS for a long time, successfully. And so now let's look how if we have all this satisfied and we are looking, let's say, at the fluorescence intensity from the observation volume of a confocal microscope, how we can derive the information we want. So again, we have this observation volume, a particle diffused through the volume, and that resulted in a burst of fluorescence intensity. Of course, if we could clearly see the individual burst, we could possibly measure the duration of the burst directly, but that's not always easy. There could be several particles, the bursts might be overlapping, and there could be quite a lot of noise. So it's not very clear where one burst starts, where it ends. So in fluorescence correlation spectroscopy, as the name suggests, we are using a different approach. We are using correlations of the signal. So let's imagine this is the fluorescence intensity time trace we have measured, and then we take this measured intensity and we are multiplying it with its replica, which we are then gradually shifting in time. We are introducing different lag time between the original time trace and the replica, and we are calculating the products. And of course, in the beginning, when they were perfectly overlapping, the product is the highest. And now that I am shifting it away, the product is getting lower, because sometimes I have a high value in the original trace, but then the value in the replica is already getting lower. And when the shift is very large, then there's no more systematic coincidence between the bursts in the original and in the replica, and the product reaches its asymptotic value, which is basically just the square of the average fluorescence intensity. So this is typically then used as the normalization factor, and this is then the autocorrelation function of the signal. Now, if we have a larger particle which diffuses slowly than the smaller particle, then the correlation function will decay at longer time because the bursts are broader. So even at larger shift, they are still overlapping. So the correlation is still high. And now if we have multiple particles on average in the observation volume, so then the average signal is proportional to n, which is the number of the particles, while each of the individual bursts is proportional to the intensity contribution of each individual particle. And from that, it follows that the amplitude of the autocorrelation function is inversely proportional to the average number of particles. So the more particles there are, the lower is the amplitude of the autocorrelation function. So for this reason, it's good to have low number of particles. So that means small observation volumes, relatively low concentrations, because if the number of particles is very high, the amplitude will be extremely low. So what happens if we have different types of particles together in the observation volume? Then, well, the correlation function is additive. So the overall correlation function will be sum of autocorrelation functions from the smaller particles and the larger particles. And we can then, from fitting the correlation function with an appropriate model, we can find out the diffusion times of the individual populations, the particle species, as well as the fraction of the larger or smaller particles. This is, of course, here you can see in the shape of the correlation function that there's clearly one more process. It's not always that obvious. This is quite an exaggerated case because remember, this is a logarithmic case, logarithmic scale. It's not a linear scale. So this means that this longer diffusion time is more than an order of magnitude longer than the shorter diffusion time. Then we can see that there's a bump in the correlation function if the difference is not that big. If it's maybe 5 times just longer, then most likely the correlation function will still appear as smooth. Only the shape will be slightly different than if there's only one population of particles. So we will not see it at first glance, and we need fitting with an appropriate model to conclude whether there's just one type of particles or multiple types. If we have not just diffusion of particles, but also blinking of the fluorophores, then that will again add to the correlation function. It will appear as an additional process in the correlation function, which has its typical duration. And luckily, in most cases, the blinking of fluorophores is quite fast. So it happens on a time scale shorter, again, significantly orders of magnitude shorter than the diffusion. So it's possible to separate it in the fits of the correlation function. If the diffusion and blinking happen on very similar time scales, then that could be difficult to separate them, separate their contributions to the autocorrelation function. Now let's consider an example of a type of study where we can use FCS, confocal FCS. So let's take, we have a suspension of liposomes, which are loaded with a fluorophore, water-soluble fluorophore. So we, by chromatography or by dialysis, we separate the fluorophore, which is not inside of the liposome. So the fluorophore is only captured inside of the liposomes. And then we add a membrane active compound, for example, some antimicrobial peptide. And we can study what happens to the liposomes. Sometimes these peptides are known to cause pores in lipid membranes. So if that happens, then the fluorophore will leak out, and we will see in FCS, we will see a change in the diffusion, the coefficient, because here the fluorophore moves slowly because the diffusion coefficient is determined by the liposome, while here it will be moving faster because it's the movement of the individual fluorophore molecules. And there will be also change in the particle number, because depending on how many fluorophores on average are encapsulated in the liposome, if we release them, then if, let's say, there would be 4 fluorophores on average in a liposome, we would expect to have approximately 4 times higher particle number after they were released out. We can make it even fancier. Let's say we add a lipophilic fluorophore to the liposome membrane, and then we can distinguish whether the fluorophore escaped by formation of pores in the liposomes, which release the fluorophore, but the liposomes still remain more or less intact, or whether the molecule acted as a sort of detergent and it dissolved the liposomes and created lipid micelles. So this is something which is an approach that has been used repeatedly in studies of membrane active compounds and how they interact with lipid membranes of different composition. Now that we are talking about using two types of fluorophores, so there's something more we can do if we have two types of fluorophores, and we can measure, of course, the signals of the two fluorophores, and we can cross-correlate them. So now we are not looking at products of the signal and its replica shifted in time. We are looking at the product of the signal in one channel with shifted signal in the other channel. And of course, if the fluorophores move independently of each other, then there's no correlation between the two time traces. So the cross-correlation amplitude, there's no cross-correlation, is flat. On the other hand, if the fluorophores move always together, so there's one particle which contains both fluorophores, then the bursts happen at the same time, and there's high cross-correlation between the two time traces. So we could use this also for example, for the liposome leakage assay, because we could also look at cross-correlation between the signal of the fluorophore entrapped inside the liposomes and the fluorophore which labels the membrane of the liposome. As long as the fluorophores are trapped inside, they should cross-correlate, and once they leak out, then the cross-correlation will be lost. And so you can now say that what I've been talking about is very old-fashioned stuff, because we're talking here about some liposomes, model systems, in vitro assays. So that's, of course, that's the type of things that I was doing when I was young. But at that time, people were saying that the cells are the cuvettes of future. So I should now say that the cells are the cuvettes of today. And for a long time, people have been using FCS for measurements inside of living cells. So it's the ideal of studying molecular processes directly in a living cell. So this is just it's from a review paper where this should show the variety of different processes in living cells that have been studied by FCS. So, for example, some people study chromatin in the nucleus by looking at how fluorophores can diffuse through chromatin. For example, Hendrik, who is here, is doing these type of things. And then people are looking at membrane receptors, transport of all sorts of things throughout the cell. Basically, whatever process you can think that you might find some way how it can be characterized using FCS, looking at changes in diffusion, mobility of molecules, concentrations, and so on. And of course, we don't necessarily stay just with live cell. We can go to any living organism, such as zebrafish embryos. But of course, there arises one problem, that cells, organisms, they are all very inhomogeneous. So let's say we have here cell with 10 micron scale bar. This is a map of Singapore with 10-kilometer scale bar. So if we use this parallel, then we probe this with a diffraction limited spot, some 200 nanometer spot. So this is like if we are probing Singapore with a 200-meter spot. So if we place it somewhere here in the central catchment, we might get the impression that the whole Singapore is a jungle, which might have been true a few hundred years ago, but it's not true anymore, as we all know. So luckily, SCELSE it's a bit better than with cities, but still the same thing applies. The cell is not necessarily a homogeneous system. So if we are looking at a single small volume, it might not be representative of the whole cell. So we ideally should probe multiple positions, but that takes time. The cell might be dead because it might not like that we are illuminating it with laser, and so it's not ideal. So we could. So the question is, can we do it in? Can we parallelize FCS? Do FCS in many positions at the same time? Ideally, do it in for a whole image in a wide field setup. Well, some putting it this way, so the obvious answer is yes, we can do it. And for example, Torsten Woland has been the pioneer of this approach of imaging FCS. So let's look if we have all that is needed. So we need single molecule sensitivity, sufficient temporal resolution. So modern cameras satisfy this. And question is, how is the small observation volume defined? So if we take a camera image, so in the lateral direction, it is defined by the pixels of the camera. And so, and if the pixels are smaller than the microscope point spread function, then of course we have to consider also the point spread function of the microscope. Typically the case that the point spread function is larger than the pixel area. So molecules which are geometrically outside of the projected pixel will still contribute to the signal. So we should consider it's the convolution of the pixel area with the microscope point spread function. And provided the pixel is very small, then the point spread function will be the dominant factor. And if the pixels are large, then the point spread function will add only marginally to that. And now how it is in the axial direction. So there, ideally should confine it somehow. So the usual approaches are either total internal reflection fluorescence, TIRF, where the axial direction, the limit is limited by the penetration depth of the evanescent field TIRF, which is very small. So this is would be less than 100 nanometers. Or if, but of course, it's limited to the surfaces. So this is ideal for cell membranes. For other situations, we could use, for example, a spin microscope, where it will be defined by the thickness of the light sheet. So can be done. So this is how an example of roughly to give you an idea of imaging FCS. So we take some noisy images and take a stack of images with high temporal resolution. So typically can take tens of thousands of frames with millisecond or a few millisecond time resolution. So this is a intensity time trace in a pixel. This is an autocorrelation function from an individual pixel. These are the autocorrelation functions of all the pixels in an image. And then if we fit them with an appropriate model, we can get maps of the parameters of interest, such as the number of particles or the diffusion coefficient. So this image is then the map of diffusion coefficient. So this is an example how this can be used. So this is example using a TERF microscope, because this study was looking at cell membrane, which was labeled by a lipophilic dye. So we are looking at diffusion of lipids in the membrane. And then the cells were treated with a polypeptide, human islet amyloid polypeptide, and a time lapse was taken, repeated imaging FCS measurements over the course of 1 hour. So you can see how the changes in the diffusion coefficient. So in the beginning, it briefly becomes faster in some places, but then as over the time, it becomes well, the final time point is when there's the most of the blue, which means the slowest diffusion. So that's the end point after incubation for 1 hour with this light. Sometimes we can, so this is, you can say now our resolution is limited by the pixel convolved with the microscope point spread function, but sometimes we can even derive some information on scales smaller than the point spread function or the pixel. So we can look at relationship between observation area and the average residence time. So if it's free diffusion, that should be proportional. But in some cases, if there are some barriers to diffusion, some obstacles, like for example, a meshwork of semi-permeable barriers. So the fluorophore can jump over the barrier, but it requires some activation energy. So it slows down the diffusion. So when we are looking at a very small area, which is in between the barriers, diffusion appears to be free. But then when we start looking at larger areas, the effective diffusion is slower because the particle needs to hop over several barriers. So then there would be such a break in the dependence, the diffusion law plot. Similarly, if there are domains which act as transient traps for the fluorophore, so when the fluorophore is in the domain, its diffusion is it cannot diffuse too much. Its diffusion is slow. It's limited by mainly by the residence time in the transient trap. And when we are looking at larger scales, then there will be the trapping time component, but then the diffusion will play a larger role. So then let's say these dashed parts of the plots are below the resolution limit. So we cannot directly see these structures. But if we extrapolate the measure, so this is what we measure when we are changing the effective observation area, and then we extrapolate it to 0. And for free diffusion, the intercept should be 0. For the transient traps, it tends to be positive. And for the meshwork, the hopping over the barriers, it tends to be negative. And how can we measure at different observation areas? Well, in imaging SCS, it's very straightforward because we can just look at several pixels together, bin the signals, and calculate the correlation function from the binned time trace. So by looking, so can think of it that by looking at larger areas, we actually can extrapolate and get information on something which is smaller than the pixel, the individual, the smallest observation area, which is achieved by the hardware. This is an example, a model systems where we can clearly see the obstacles because they are large enough, larger than the resolution. So we see here the pattern, which is just a lipid bilayer on a patterned surface. Here there are large domains of lipids which are in a gel phase lipids, and of course that is just to sort of proof of concept that we can see that in the homogeneous bilayer the intercept is 0. In the case of domains, it's positive. In the case of the pattern, it's negative, and we can even see here the break in the diffusion law because we can directly measure smaller areas than what is the pattern. But we can, of course, then use this in a more interesting system, for example, in a zebrafish embryo. And it was shown here by imaging FCS that this morphogene tends to partition into cholesterol containing domains in cell membranes. We can do also fluorescence cross-correlation spectroscopy in an imaging format. So we take 2 images in 2 channels. So this is taken by the same camera, just the image is split onto 2 halves of the camera. And then we get 2 autocorrelation functions and 1 cross-correlation function in each pixel. And we can get the map of diffusion, the coefficient for each channels, and the maps of the particle numbers. And from the particle numbers, we can then find the cross-correlation amount map, which is a measure how strongly cross-correlated are the signals. So that can be, for example, an indication if we label 2 different proteins, one by green, one by red fluorophore, then we can see how, if these proteins interact together, if they form complexes, how strongly they form the complexes. Again, example, this is a study of EGFR dimerization. So we have 2, we start with a negative control, which is which are two proteins which are not supposed to interact, labeled by green and red fluorescent proteins. So we have low cross-correlation amount. Positive control is a single protein labeled by both fluorescent proteins. So we have high cross-correlation amount. Of course, you can say that it's not that high. You would expect it to be higher in this case. Why it's just around 50%. And the reason is that in the case of fluorescent protein, this is something which is expected because fluorescent proteins are not all the all molecules of fluorescent protein are fluorescent. So there's quite a substantial amount of non-fluorescent fluorescent protein molecules. You know what I mean? So those proteins will appear just a single labeled. And that's the actual experiment where we have a population of EGFR molecules labeled by GFP, population labeled by MRFP, and if they form a dimer, then we should see positive cross correlation, and we see quite high cross correlation. Sometimes it's very high, so that's probably even higher complexes than dimers, maybe tetramers or higher oligomers. And we and since we have now all these pixels, we don't need to do just cross correlation between two different fluorescence channels. We can also cross correlate signals between two different pixels. So here are autocorrelation function. Here we correlate this pixel with this pixel. I mean, it looks quite similar, but if we go a bit further, then you can see the difference, because the amplitude is shifted from 0, because it takes some time for the molecules to diffuse from this pixel to the other one. And if this can be quite interesting if there are any barriers to diffusion, such as exemplified by this barrier from real life, macroscopic life in Berlin in the past. And then we could see cross correlation if the pixels are on the same side of the barrier, but if they are on the opposite sides, then we shouldn't see the cross correlation. And if it's a barrier which allows transport only in one direction, then depending on which way we calculate the cross correlation, we might see cross correlation or no cross correlation. So there's quite a lot of things that can be done. So in a way, these data sets which are used for imaging FCS are very have a high information content, because there are images, microscopy images which have quite high spatial resolution, and there are stacks of such images acquired with high temporal resolution. So that's quite a lot of information there. And FCS, the correlations are just one way how to extract the information from all the fluctuations and the noise which is contained there. So now recent work done in Thorsten's lab shows that deep learning might be even better in some cases than the correlations in extracting the information from the noisy images, noisy image stacks. Now, last question, what if we don't have sufficient temporal resolution? So just before we say bad luck, but let's first consider what does the temporal resolution mean. So let's say in the camera stack, which we use in imaging FCS, so we have some camera exposure, then there is some time needed for readout of the camera chip, and together the exposure and the readout time, that is what determines the temporal resolution. And that's something that ought to be much shorter than the duration of the burst, so that we can see the correlation. Now, in some cases, we might have a very short exposure, but a long time in between. So this is, for example, the case of laser scanning confocal microscope, where we obtain the image by scanning. So each pixel is exposed for a very short time, but then we need to wait for all the other pixels to be scanned before the beam returns back to the same pixel. So there's a long time in between, very short exposures. Now, this is something which you might be able to work with. course, with some limitations. So the easiest way, we can say, okay, we forgot about the mobility because we cannot sample the correlation. We are just basically getting isolated samples, so we cannot measure the duration of the burst. We are just randomly sampling the fluctuating time trace. And if we look at the mean and the variance of such a time trace, we can actually derive information on the concentration, and from that on brightness of the molecules. And this is an approach which is called number and brightness analysis. So that doesn't require the high temporal resolution in this sense like we need for imaging SCS, but it just needs the exposure needs to be sufficiently short that you don't average the fluctuations within the exposure time. So that's one quite easy way to utilize the fluctuations. And another thing, if let's say we are using the laser scanning microscope, so we are scanning here. So we have different pixels. Say this is the direction of the scanning, and each pixel is acquired at slightly different time. So if we have the particle, there's a point spread function sitting particle is the center of the point spread function. What if the particle diffuses somehow in this direction? Then by correlating signal from this pixel and this pixel, or this pixel with this pixel, then we are basically getting the signal about information about the particles at different times, which are this very high temporal resolution, which is very high because the pixel dwell time is very short. We are just not looking at the same point in space, but we are looking at slightly shifted time, but also we are slightly shifted in space. So instead of temporal correlation function, we are getting something which is we can call a spatial cross correlation function. So instead of the lag time tau, we have some lag distance. And so this, if we do it just in one direction, that would be line scan SCS. And if we do it for a whole image, like the usual confocal laser scanning microscope image, then we have also we can also correlate along the other direction. So this is we are scanning, this is the line, and then we would be correlating the pixels in different lines. And then we get a spatial correlation function. And this is an approach which is called raster image correlation spectroscopy. So the difference from imaging SCS is that we don't need the high temporal resolution of the whole images. So this can be done with a laser scanning confocal microscope. But the price we pay is that because we get from the whole image, we get one spatial temporal correlation function. So we get one diffusion coefficient from the whole image. So we are sacrificing the pixel-wise spatial resolution of imaging SCS. But still, it's a powerful and interesting FCS mobility. So that's all from me. And of course, most of the work I've been talking about is related to what I was doing in Thorsten Wohlang's lab at NUS. And thank you for your attention. And now it's time for your questions.
Hi, Radek. Can you hear me?
Hi, Alesh.
Hi, Alex. Yes, one question. Do you have any users in your facility that are using any of these methods?
Sadly, not at the moment. I hope we might have some in the future. So that's also one of the reasons why I am talking about it now, to promote these techniques, to make people aware of what they can do. And maybe if someone gets interested, then can come to us and we can look what could be done for them.
Yeah, and do you think it's what is causing this lack of users in this field? Do you think it's because the methods are not known, they are too complex, or there are no biological questions currently that the groups would have? Just curious about this.
Yeah, I think there's not just one reason. I think it's an interplay of all the reasons you mentioned. So one thing could be that people are not that well aware of it, and then when they are designing their studies, then maybe they don't think along these lines, so they don't formulate the question in the way that would lead them to these techniques, to be answered by these techniques. And of course, the complexity could be a part of it, but that's in a way the reason why we are here. So if people are interested in that, we can help them. Of course, sometimes could be that one, of course, has to be prepared that to sometimes to get a good FCS study working, it needs some time to tune the way how the constructs you use, the way how you do the experiments. So there has to be some effort put in the beginning, and the first experiments might not be immediate success. So that could be also that sometimes people are afraid of this and don't want to step out into the unknown and stay with what they think is familiar to them and easy for them to do, and where they expect less obstacles.
And have you tried, when you sometimes spotted a potential project that could potentially benefit to, for example, measure like illegally some FCS data on with the user sample and tell him, okay, or her, you see, this is your imaging, but we can provide extra information. So now you know the concentration. Now you know how fast does it diffuse. Because this is sometimes they're trying, then the people are bringing, they just want to get the images, but we force them to acquire some extra data and try to persuade them that they get more information.
I haven't got to this, but yeah, I think this is a possibly a good strategy. So I hope I can identify some such case where maybe we can use this and move towards these techniques.
Yes, and if I can have one technical question, because for the cross correlation, you mentioned that you got 50%, which is actually pretty nice. I do agree with that. So what's the highest cross correlation coefficients for biological molecules like labeled with fluorescence proteins you have ever observed, we have ever seen?
So I mean, for the fluorescent proteins, yeah, the around 50% I think is the maximum, at least for the ones that I was using. There might be, so mainly the limiting factor here was the red fluorescent protein. The green fluorescent protein is pretty much, most of it is fluorescent. So there might, if you choose some better fluorescent protein, and maybe there are nowadays some better fluorescent proteins also in read. I'm not that familiar with all that's available. Maybe Yongvi would know better, because this was it was actually mainly his topic. There's this nice paper by Yongvi where he studied this at that time for M-Cherry and MRFP with GFP, and what is the maximum cross correlation that could be obtained. So maybe with some other proteins one can get better.
Yeah. Okay. Thank you for that. Clearly, because there's the common question people are asking: what fluorescent pair shall we use for FRAC, for cross correlations and these things? So this is a very common question from the users. So probably we should get more information in this field as well. What's currently on the market?
Yongvi is the best person to talk to about this.
Well, I haven't been doing much of the fluorescent proteins for the past, I don't know, 5 years. The paper that I published was more than 10 years back. So that was the M-Cherry and the MRFP, when both of these red fluorescent proteins are probably one of the best in the market at that time. But currently there's like lots of other red fluorescent proteins. Yeah, so I haven't been keeping up to date with the fluorescent proteins as I have now shifted to some other kinds of different types of work. But what I can add on is, if you are not using fluorescent proteins, for example, if you are using fluorescent dyes, the cross correlation can be very, very high. I have tried using oligos, so complementary oligos, one with Alexa 488 and the other one with Alexa, I think 568. The cross correlation can reach up to about 90+%, which is very, 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 very high compared to fluorescent proteins.
Yeah, this 50% is applied to fluorescent proteins with fluorophores. I mean, then again, then it would be limited by how well you can label it. If you have any, like, what is your labeling efficiency? Because in this case, all the fluorophores would be fluorescent. It won't be like the fluorescent proteins, which they don't fold properly, so they are not fluorescent and things like that. But with the fluorophore, it would be then limited by what is your labeling efficiency. If you have the target molecule, which the fluorophore got hydrolyzed away, and that would be reducing the cross correlation. If you have very fresh, and you can assume it's close to 100% label, then you can get very close to 100% cross correlation.
Everyone hear me? This is Yang Chen. Okay, so could you give a bit more detail about the use of deep learning in FCCS? Because I was wondering if we actually train the models with the FCCS or FCS data, and would that actually be time consuming? And if we actually have to, say, train a different model for each situation that we have, would that be very labor intensive also? So yeah, in that sense, would it actually be sort of quite difficult to use it in practice?
Well, this is a very early stage work, so I think at this stage is more like probing the concept, and it was done. They've been trying slightly different approaches, but the idea is that you have the data, the sort of fluctuate image series with the fluctuations, and they simulated these data. So we use a sort of Monte Carlo simulation of diffusing particles, and that produces the fluctuating image. With you input certain diffusion coefficient which you want to simulate, and then you produce many such simulated images, and you train the network on that. And of course, with the simulated images, it was very successful in getting out I mean, not just the training set, but other randomly simulated images. Of the same time, it was very successful in getting the correct parameters out. For the lipid bilayers, it was slightly less successful. And yes, you are right. So if it was to go to other systems like live cell, then you would have to consider, especially where you don't have free diffusion, but you could have things like any obstacles, any domains. So you ideally should train it on a data which simulate all this. So then it might become, might not be very feasible to simulate all the possible scenarios. I think one would have to see how specific you need the training system needs to be. In a way, this is a bit similar to the approaches which people are now doing as an alternative to deconvolution, where they have this. They also train network on simulated images, where they have a simulated ground truth, and they simulate the convolution with point spread function, and then train the network. So again, ideally you should train it on the same type of structures which you expect in the real image, compared to the classical deconvolution, which is in a way agnostic of the structures and doesn't matter what the structures are, because this is using sort of first principles. So I would say, in an analogy, correlations are like the standard deconvolution, while the analysis using deep learning would be something similar to these approaches. And it turns out that some of these approaches work quite well, even on images which are not exactly too similar to what they train them on. So I don't know. I think it's still something that has to be further explored to know whether it will be practically usable or not. In the meantime, correlations are the proven approach and the standard.
Okay, thank you.
Okay.
Hi, Radek.
Hi, Hendrik.
I was wondering what kind of FCS instrumentation do you have at your site?
Well, we are not really strong here on FCS instrumentation. We have a confocal microscope with which we can do FCS, and of course we could do things like RIGs with that as well. And we have the same setup also contains turf. So in principle, we could do imaging FCS, but it's not not optimized for imaging FCS, I would say. So maybe in the future we might have some better equipment. Also, if there's more demand for these techniques, this might go hand in hand.
Okay, thank you, Radek.
So if there are no other questions, then thank you all for participating, and I'll be looking forward to seeing a lot of you next month.
Okay, thank you. Bye bye.